Hello everyone and a very warm welcome back to MooTechie. It's wonderful to have you with us today as we explore a topic that is both incredibly common and vitally important, the microbiology of the fruits and vegetables that fill our plates. We'll uncover the hidden microbial world that leads to spoilage and learn how to protect these nutritious foods. Let's get started. Now, let's begin our presentation. Microbiology of fruits and vegetables, source of contamination, spoilage and prevention. Fruits and vegetables are a rich source of energy, bodybuilding nutrients, vitamins and minerals. Their nutritional value is highest when they are fresh, but we can't always consume them instantly. During the harvest season, fresh produce is available in plenty, but at other times it can be scarce. Moreover, most fruits and vegetables are only edible for a very short time unless they are promptly and properly preserved. One key characteristic is that fruits and vegetables have a high water activity compared to meat, fish, and cereal foods. This makes them more susceptible to microbial growth. Many of these products are protected mechanically by pectin, which acts like a protective gum between cells and gives them their firmness. However, fruits and vegetables also have a much lower concentration of free amino acids and other nutrients than meat, fish, and milk. This changes the spoilage dynamics. As fruits and berries ripen, their own endogenous pectinases start to hydrolyze the pectin. This natural process also makes the products more vulnerable to microbial attacks. Spoilage often starts with the hydrolysis of this pectin. Once pectinases damage the structure, other organisms join in, leading to soft rot. This spoilage usually occurs during storage and transport, while the produce is waiting to be processed. From the moment they are harvested, they are subject to contamination. During transportation, mechanical damage can increase susceptibility to decay, allowing microorganisms to grow. Pre-cooling and refrigeration during transit can slow this growth down. Washing the produce might seem helpful, but it can actually distribute spoilage organisms from damaged items to whole ones. And if the water is recirculated or reused, it's likely to add even more organisms. So where does this contamination come from? Sorting spoiled fruits or trimming bad parts removes some microbes, but the additional handling can cause mechanical damage, making the produce even more susceptible. In the processing plant, fruits and vegetables face further contamination. Some procedures like adequate washing, steam peeling, and blanching reduce microbial numbers. But others, like sweating of products during handling, can increase them. Bacterial counts on fresh vegetables arriving at a plant can range from 10 to the power of 2 to 10 to the power of 7 per gram, depending on the species and condition. Hot water blanching reduces total organisms but may cause a buildup of spores from thermophilic bacteria which can later spoil canned foods. A major concern is the buildup of microorganism populations on equipment from exudates and residues. This can greatly influence food contamination. Including decayed parts in fruit juices also drastically increases microbial numbers. Now let's talk about the spoilage itself. The deterioration of raw vegetables and fruits can result from physical factors, the action of their own enzymes, microbial action, or a combination of these. Mechanical damage from animals and birds predisposes the produce to increased enzymatic action and microbial entrance. Contact with spoiling items can transfer organisms, causing spoilage to spread and increasing wastage. Diseases from non-pathogenic causes like brown heart disease in apples or black heart in potatoes also end with spoiled products. The predominant type of microbial spoilage varies not only with the kind of fruit or vegetable but also with the variety. It can be due to plant pathogens or saprophytic organisms acting as secondary invaders. Here are some of the most common types of spoilage. First, bacterial soft rot caused by Erwinia carotovora, which ferments pectins. It results in a water-soaked appearance, a soft, mushy consistency, and often a bad odor. Gray mold rot, caused by Botrytis cinerea, is favored by high humidity and warm temperatures. Rhizopus soft rot, caused by Rhizopus stolonifer, also results in soft and mushy food, often covered by a cottony mold growth. Anthracnose, caused by Colectotrichum lindemuthianum, appears as spots on leaves and fruit. Alternaria rot, caused by A. tenuis, appears as greenish-brown areas that turn brown or black. Blue mold rot, from Penicillium digitatum, shows a characteristic bluish-green color. Downy mildew, caused by molds like Phytophthora, grows in white, woolly masses. Watery soft rot is found mostly in vegetables and is caused by Sclerotinia sclerotiorum. Stem end rots involve the stem ends of fruits and are caused by genera like Diplodia and Fusarium. Black mold rot, from Aspergillus niger, shows dark brown to black spore masses. Other types include black rot, pink mold rot, fusarium rots, green mold rot, brown rot, and sliminess or souring from saprophytic bacteria. Fungal spoilage of vegetables often results in water-soaked mushy areas. In fleshy fruits, rots often show brown or cream-colored areas with mold growing below the skin. Some fungal spoilage appears as dry rots, where the area is dry, hard, and discolored. Now, let's consider fruit and vegetable juices. Juices can be squeezed directly or obtained by maceration. They are acidic, with a pH from 2.4 to 4.2, and contain sugars from 2% in lemon juice to 17% in grape juice. Exposure to air and high moisture content favors spoilage by fast-growing yeasts and bacteria. The deficiency of B vitamins discourages some bacteria. Storage temperature is a critical factor determining which organisms dominate. At room temperature, the normal change in raw fruit juice is an alcoholic fermentation by yeasts. This can be followed by oxidation by film yeasts or molds if exposed to air, or the oxidation of alcohol to acetic acid by acetic acid bacteria. Other spoilage in juices includes lactic acid fermentation by bacteria like Lactobacillus pastorianus, fermentation of organic acids, and slime formation by leuconostoc. Vegetable juices are less acidic, with a pH of 5.0 to 5.8. They contain more growth factors and support the growth of fastidious lactic acid bacteria, leading to acid fermentation. When juices are concentrated, the increased acidity and sugar favor the growth of yeast and acid-tolerant bacteria like leuconostoc and lactobacillus. It's also important to note that spoiled fruits and vegetables can cause various diseases including salmonellosis, shigellosis, Campylobacter enteritis, yersiniosis, listeriosis, botulism, and Norwalk-like gastroenteritis. Now, how do we preserve these precious foods? Let's start with vegetables. A primary method is the use of low temperatures or chilling. Most vegetables preserved without special processing are cooled promptly and kept at chilling temperatures. This is done using cold water, ice, mechanical refrigeration, or vacuum cooling. Pre-cooling immediately after harvest, often with a cold water spray called hydrocooling gray, is very effective. Each vegetable has its own optimal temperature and relative humidity for storage. For example, ordinary potatoes turn sweet below 2.2 to 4.4 degrees Celsius and are stored at higher temperatures if meant for chips. Freezing is another powerful tool. Washing reduces some microbes, and scalding or blanching at 86 to 98 degrees Celsius brings a great reduction, as much as 90 to 99%. The freezing process itself kills about half of the remaining organisms, and numbers continue to decrease during frozen storage. The types of bacteria that grow upon thawing depend on the temperature and elapsed time. At fairly low temperatures, Micrococcus species might predominate. At higher temperatures, Flavobacterium may multiply. A critical safety note: cooking frozen vegetables will not kill all spores of Clostridium botulinum. Therefore, cooked frozen food should not be left standing at room temperature for extended periods. Drying is also used for vegetables, especially in soups, spices, and condiments. Methods like explosive puffing create a porous structure that helps with reconstitution. Drying by heat destroys yeasts and most bacteria, but bacterial spores and heat-resistant vegetative cells usually survive. Microbial counts on dried vegetables are usually higher than on dried fruits because vegetables are generally less acidic. Regarding preservatives, sodium chloride is the most common added chemical. In sauerkraut, 2.25 to 2.5% salt is added. Vegetables high in protein like peas and beans can be preserved with enough salt. In salad bars, sulfites are sometimes used as salad fresheners to prevent enzymatic browning, but these residues have been associated with asthmatic attacks. Acid fermentation in chopped vegetables by lactic acid bacteria creates a pleasant flavor. Salt is added to inhibit the growth of undesirable contaminating bacteria. The amount of sugar determines the final acidity, and the salt and temperature control the rate of acid production and the types of bacteria involved. Finally, let's look at preserving fruits and fruit products. The surface of healthy fruits is dominated by yeasts and molds. Affected fruits are sorted, and spoiled portions are trimmed. The interior of healthy fruits typically has very few microorganisms. Fruits are grouped by their pH into acid foods like tomatoes and pears and high acid foods like berries. Washing reduces contaminating bacteria. Preservation methods include chilling, sometimes with added chemicals like hypochlorites or propionates. Waxes and mineral oils can be applied to mechanically prevent the entry of spoilage microbes. Controlled or modified atmosphere storage is also used. Freezing extends shelf life but can cause damage, resulting in flabbiness and juice release. Yeasts like Saccharomyces and molds like Aspergillus survive freezing, with yeasts more likely to grow during slow thawing. The natural flora before freezing is often dominated by coliforms, enterococci, and lactic acid bacteria. Among coliforms, the Enterobacter aerogenes type is part of the natural flora of fresh and frozen fruits. Drying is also a reliable method to extend the storage life of fruits. And that brings us to the end of our presentation. Thank you. We've journeyed through the entire life of fruits and vegetables, from the field to your table, exploring the invisible microbes that threaten their quality and the clever methods we use to fight back. From the initial breakdown of pectin to the final stages of soft rot, and from simple chilling to advanced controlled atmosphere storage, understanding this microbial interplay is key to reducing waste and ensuring food safety. Thank you for your time and curiosity. We hope this session was fruitful, pun intended. We look forward to seeing you in the next class.